Hey, Assalamu alaikum. In this video, we will understand about the cardioprotective signaling pathway, risk and safe pathway. So basically, it is activated during the ischemic preconditioning whenever the arteriosclerosis or the atherosclerosis or other problem or other hypoxic condition due to the less infusion of the oxygen and the lungs injury, so or other form of stress. So in this way, the subsequent ischemic injury will lead to activate the two pathway, perfusion injury salvage kinase pathway. This is known as the risk, and the survival activating factor enhancement pathway, which that is the safe pathway. So in this way, both pathway is important for the survival of the cell, not necrotic triggering. So the ischemia due to blockage of the artery, which that is the coronary artery can be. And in this way, remember the cardiomyocytes survive due to this injury or due to this disease. So in this way, the blood impede to the coronary artery. So in this way, not provide the oxygen to the cardiomyocytes of the basically the heart tissue, the cardiac tissue or cardiac muscle. So the hypoxic shock will occur of the cardiomyocyte and decline muscle contraction due to the less oxygen. In this way, the risk and safe pathway will trigger. Risk and safe pathway trigger also different things like adenosine, bradykinin, opioids, angiotensin 2 and growth factors. Mechanical stress and hypoxia also due to the ischemia or something. In this way, the safe pathway basically the hypoxia and the pharmacological agents will trigger this say risk pathway, but the safe pathway due to the interleukin 6, TNF alpha, epoprotein, erythropoietin, granulocyte colony stimulating factors, and hepatocyte growth factor, as well as stress signal. So in this way, here I will draw the bigger picture of the cell to understand where is the signal where is the pathway which that inhibit the apoptosis of the cell or necrosis of the cell. So collectively we will understand. So here is the safe pathway and the risk pathway we will discuss. So here is the cell which that show basically this is the G coupled protein receptor contain GI protein and GS protein. So whenever the ligand will bind or the hypoxic condition of the cell will trigger certain type of the growth factor. And in this, the GI and the GS collectively trigger the phosphorylation, a cascade of phosphorylation, the PI3K pathway activation, AKT, and the P70S6 kinase pathway will inhibit the glycogen synthase kinase 3 beta. This glycogen synthase kinase 3 beta is responsible for the triggering of the MPTP channel for the outflux of the cytochrome. When outflux of the cytochrome, in this way the apoptosis will trigger. So we should inhibit it. So the GS, glycogen synthase kinase 3 beta inhibition due to the protein kinase activation through the GS protein will activate the endothelial nitric oxide synthase enzyme, lead to vasodilation also occur for the providing of the oxygen again, and also the endothelial nitric oxide activate the PKG protein kinase G protein will inhibit the glycogen synthase kinase 3 beta. This glycogen synthase kinase beta also when inhibit, so the MPTP will be inhibit ultimately, so not release the cytochrome C, so not apoptosis. While also the reactive oxygen species outflux from the mitochondria from the safe pathway. This is the risk pathway. So will lead to prevent to damage due to the protein kinase A pathway activation. This endoplasmic reticulum contains silica as a channel protein for influx of the calcium. So silica will inhibit. So silica protein inhibit and influx of the calcium will not occur. Why? Because the PLN protein, phospholamban formation. So the phospholamban will not occur. So dephosphorylation. So here is the T3 due to mitochondria and sarcoplasmic reticulum due to the dephosphorylation MFN1 and 2 will not so it is occur. So in this the PLN phosphorylation to dephosphorylation will occur due to the PLN activation to inhibit the calcium uptake from the endoplasmic reticulum. And in this way the PLN inhibits so the circuit for uptake calcium so inhibit, and the PNL phosphorylation not inhibit. So in this way, dephosphorylation will occur, lead to ryanodine receptor 2 will not pump out the calcium due to the not excessive calcium. If the excessive calcium, so it can move into the mitochondria, lead to increase the calcium overload. So the overload due to the MCU channel, when calcium will increase, will lead to damage the mitochondria. So it will not occur in the risk pathway. But the safe pathway is the GP130 protein and the TNFR1 as a extracellular domain and intracellular domain phosphorylation. Whenever the signal, stress signal will bind, will lead to activate the JAK and STAT3 signaling pathway, will inhibit the MPTP. When inhibit the MPTP, lead to not outflux of the cytochrome, and in this way the apoptosis will not possible, because the cytochrome is very important and essential for the apoptosis. And also the reactive oxygen species move out via a potassium reactive potassium Ross exchange channel MKTP. MCATP. So MCATP is important for outflux of the reactive oxygen species, and the reactive oxygen species again trigger the risk pathway due to the pKa and endothelial nitric oxide synthase activation. So same inhibit the glycogen synthase kinase 3 beta. Inhibition will lead to allow the MPTP to outflux of the reactive oxygen species. And in this way, here you can see the ROS will prevent the damage of the mitochondria due to the outflux, and the potassium will inside. Connexin is the important protein also present in the plasma membrane. Connexin will recruit toward the mitochondria, bind with the intra-mitochondrial membrane. Connexin 43 will trigger the electron transport chain efficiently. So in this way, the work normally. And also the MKATP trigger for the outflux of the ROS. So the safe pathway go directly to mitochondria, prevent critical pores MPTP, and reduce the ROS in mitochondria, and outflux increase. Risk pathway, ROS outflux, and AKT-PKC improve cell survival and improve energy production and prevent cell death signaling due to the risk pathway. Why? Because the mitochondria will not damage, mitochondrial cytochrome C will not outflux, and in this way the apoptosis will not trigger and necrosis will not trigger. So I hope you make sense about that. This is the cardioprotective mechanism as a signaling pathway we have discussed. So both risk and safe pathway important for the mitochondrial calcium overload. So decrease due to the calcium outflux, and in this way remember, so the calcium will not release whenever the PLN will inhibit the circa. So the delay MPTP opening promotes cell and survival signaling. So remember, one important thing is that the sarcoplasmic reticulum will inhibit the allow to influx of the calcium due to the PLN activation. So in this way the calcium overload into the mitochondria will not occur. So I hope you make sense about that. Bye.